Hello, everyone. Welcome to the Kirby seminar today. Before we begin, I'd like to acknowledge the traditional custodians of the land we stand on today and pay my respects to elders past, present, and emerging. So it's my pleasure today to introduce Dr. Braulio Valencia. By the way, my name is Chaturaka Rodrigo. I'm from the Viral Immunology Systems program. So I was a joint primary supervisor of Braulio's PhD. So Braulio is from Cusco, the ancient city of Cusco in Peru. And he did his undergraduate and postgraduate studies in Lima, Peru, and was practicing as an infectious diseases physician when he came to us on a science share scholarship. So his research interest while in Peru was in mucocutaneous leishmaniasis, a parasitic infection, very common in that part of the world. And after coming here, we made him work on viruses. And he took that challenge very well. And he actually, both him and us as supervisors had a very steep learning curve because he decided to do his work on human genetics and the interface of viruses and human genetics. And neither me nor Andrew are experts in that. So he actually led this whole project by himself. So as with any PhD, he has its ups and downs and a nail-biting finish. And eventually he emerged with very good outcomes. So it's my pleasure to introduce Braulio to tell us what he did during his PhD.
Thanks a lot, Chaturaka, for that kind introduction. I also like to start by acknowledging the digital people and pay my respect to the past and present, to the elders past and present. It is a great feeling to be here today. I remember during the past 5 years, I've been attending similar seminars with a lot of respect and hope, but also some anxiety and fear because my progress was not as expected at some points, but I think this is the journey any PhD student usually face. So yeah, thanks again for the opportunity. Right. So my background is basically a clinician, and in the hospital environment, we are used to deal with diagnosis issues or treatment issues. But some of the features from infectious diseases, that was very interesting for me was the diversity of clinical manifestations, but also treatment responses. In hospitals, we are used to deal with the typical, but firstly, we are not used to deal with subclinical manifestations. And this first picture is something that can look completely unusual, but it is actually a parasitic infection that Chaturaka referred before. This picture here is the classic picture of a cutaneous ulcer. But something that was really interesting for me was trying to understand why some individuals can have these very diverse clinical manifestations caused by the same pathogen. And also was very interesting to find why some individuals can respond very effectively to treatments, but some are progressing or are relapsing again and again after several treatments, or in some cases, very infrequent, progressing to very severe manifestations affecting diverse parts of the human body, as the case of these severe mutilating lesions in tegumентary leishmaniasis. So this motivated me a lot before moving to Australia to explore what is the reason why the same pathogen is causing this diversity of manifestations. And actually, during my clinical training, I was able to identify, but the same clinical the same variability in symptoms or manifestations are common in other infections. This is, for example, yellow fever. Most of generally assumed as a lethal infection, but this gentleman died due to a severe hemorrhagic infection. But his partner survived without any symptoms. So it is strikingly interesting why some individuals can have very diverse manifestations to viral infections as yellow fever, fungal infections as chromoblastomycosis, the one here, mycobacterial infections as Buruli ulcer, or parasitic infections as cutaneous or tegumentary leishmaniasis. So despite most of my interest at the beginning or before moving to the PhD was focused on neglected tropical diseases, independently of the pathogen, there are many similarities. And I think this is well explained by this graphic, in which you can identify that clinical manifestations are not solely explained by pathogen determinants. They are strongly influenced by the human, but also all the factors or many environmental factors affecting the expression of clinical manifestations. Many of these severe manifestations are in fact explained by well-characterized features. And we know, for example, that age of infection or comorbidities, co-infections, nutritional status, or gender are factors, or anatomic defects are associated with the risk of severe manifestations. But in fact, there is a proportion of individuals without any of these factors that are having severe outcomes. And in my head, in the head of a clinician that wasn't exposed to genetics before, I assume that the human genetics is probably a reason affecting this variety of in clinical manifestations. Unfortunately, genetics up to now, the impact of genetics in the severity of infectious diseases is mostly restricted to the upper spectrum of the disease that I'm showing you now. So most of the deadly manifestations or severe manifestations are explained by monogenic conditions, infrequent genetic variations predisposing to severe manifestations. And we are plenty of examples like that. For example, mutations in the complement pathways, increasing the risk of severe invasive meningococcal infections, or severe human papilloma virus infections. So genetic factors are definitely explaining this spectrum of diseases, but are not actually explaining most of the burden of infectious diseases, which are those individuals with minimal symptoms or even with subclinical manifestations, or those individuals that, due to the mild clinical manifestations, are not requesting medical support, and consequently, as clinicians, we are not having a real understanding of what is happening with them. Some interesting fact is also that the same pathogen, as I previously mentioned, the same pathogen can cause diverse clinical manifestations. And from an immunological perspective, this can be explained either in the case of tuberculosis, for example. The first two pictures are cavitating and amyloid tuberculosis, or mucocutaneous and visceral leishmaniasis. These completely opposite manifestations can be caused by the same pathogens. But the reason why the same pathogen can cause these diverse manifestations is probably because some individuals have a predisposition to mount an excessive pro-inflammatory response, or in the opposite way, a weak inflammatory response. And that's the reason why, for example, individuals without any comorbid condition explaining miliary tuberculosis can develop this disseminated disease without any comorbidity, but because there's any anti-inflammatory immunological profile facilitating the dissemination of the parasite. And in the other way, an excessive inflammatory response can cause damage, even if the pathogen burden is relatively low, or is not explaining the severity of symptoms. So I think this is a general background just to give you a great picture of how infectious diseases, irrespectively of their taxonomy or of their classification, are behaving in a very similar way. And I just want to give you a few key points, at least these were key for me while while performing my literature review. So the key messages are, and I hope you find them useful. Three concepts. So the genetic architecture of a trait. I'm going to explain you more in detail later. What available genetic or genomic methods are to study the contribution of genetics to disease severity, and an accurate trait characterization. So in terms of the genetic architecture, I think you can think in trait as many things. For example, color of eyes is one trait, or your height is a trait, but also immune responses are traits, and also or risk to suffer specific diseases are also traits. So if we have a trait, we definitely have genes explaining these traits. And from a very classic genetic perspective, we can explain traits based on the number of genes causing this trait, but also the penetrance or the effect of these mutations in causing this trait. So in general terms, monogenic conditions or Mendelian conditions, as the ones that I explained you before, those causing unusual severe manifestations or deadly manifestations, are generally monogenic conditions, which means that only one gene is enough to predispose individuals to suffer these severe manifestations, while more common manifestations, including, for example, the risk to suffer cancer or a heart attack, or even the risk to suffer COVID, are determined by several genes interacting in a synergistic way, but requiring the environment to explain the whole severity. So in this case, for example, for the risk for hypertension, at least 30 or 50 genes have been described as affecting the risk of hypertension. Only one gene or only one variation is not explaining the risk of suffering hypertension. And this is probably, and this spectrum of genetic manifestations are probably explaining most of the human diseases. There is a gray area between the monogenic and the polygenic conditions, which are the oligogenic conditions or rare monogenic conditions, pleiotropic monogenic conditions, or monogenic conditions susceptible of environmental factors. And we are discovering how these genetic architectures are explaining diseases, mostly because genetics are being included in understanding human diseases more frequently than before. So it is good to have genomic or genetic characterization of different diseases. In case of infectious diseases, the behavior is pretty similar. So primary immunodeficiencies are generally explained by a single mutation predisposing to the risk of multiple infections, while the risk of severe flu or the risk of severe COVID can be explained by multiple genes interacting and not necessarily explaining the risk with 100% of certainty. And I think this is very important, as in the picture that I show you, the iceberg picture that I showed you before. Maybe it's very in a hospital environment, it's very useful to understand why severe manifestations can be caused, or what is the genetic contribution to severe manifestations, but our understanding of the burden of disease, which is the mildly symptomatic or the asymptomatic spectrum, is not completely understood. So in terms of genomic methods, I think these 4 methods are commonly used to characterize the genetic architecture of a trait. And if you use the term trait to associate with severity or disease manifestations, we can use any of these methods to characterize disease severity. These include the simple approaches as a candidate gene approach, which is basically genotyping mutations in a single gene, through more expensive holistic approaches, which are characterizing the entire genome or multiple genes in the human genome. There are obviously advantages and disadvantages of using one or the other. But in general, to decide which method you are using depends on what is your research question and what preliminary data is available. So for example, if you don't have any preliminary data mapping or associating a gene with the risk of a severe manifestation, it is probably more useful to use a more an open-ended approach as a genome-wide association study or a whole genome or whole exome sequencing approach, as it will search through the entire human genome for genetic associations with the trait of interest. While if you have preliminary data associating a particular SNP with the severity of a disease, for example, a mutation in TNF alpha, and was already associated with disease severity in dengue, it is totally reasonable to just genotype that SNP in a cohort to validate your findings. Unfortunately, these genomic or genetic approaches are just exploring the host dimension, but are not tackling the pathogen part. So to really understand what is explaining the severity of diseases, we definitely need to analyze the pathogen contribution, but also the human contribution. And the previous methods that I described are basically tackling the genomic, the host determinants, host genetic determinants of disease, but are not tackling the pathogen determinants of disease. So as I mentioned, these methods are tackling this the host to illness phenotype, but are not tackling the effect of pathogen diversity in illness severity, or even how or genomes are affecting the pathogen diversity. So to have a real understanding of what is explaining severity of disease, we definitely need to include at least these 2 components in the equation to really understand what is driving severity. Okay, and I think the last point in this chapter is regarding trait characterization. Again, trait is anything we can identify in patients. And in this case, for example, dengue, the diagnosis of dengue or the definition of dengue severity is based on, as in other diseases, in clinical criteria. So you have clinical criteria to define pneumonia, or you have clinical criteria to define dengue or severe dengue. This is the WHO classification for dengue severity. And despite some of the clinical criteria are very objective, there are, as for example, abdominal pain or vomiting or bleeding. Some of these manifestations are not necessarily result of the disease itself, as for example, clinical fluid accumulation or liver enlargement, because they can depend on the subjectivity, for example, of the clinician assessing, making the clinical examination, or the subjectivity of the sonographies looking for ascites or a pleural effusion. And that is really important because at the end, clinical criteria is very useful to diagnose conditions or classify conditions, but it's not necessarily informing what the biological basis of diseases are. And in many occasions, clinical diagnoses are mixed with other factors, external factors that are not really explaining the disease. One way to overcome this issue is to using an alternative phenotype characterization. And as you can see here, pathophenotypes or clinical diagnosis or external phenotypes are basically what clinicians are identifying after a physical examination or a laboratory examination. But there are other factors, there are other biological mechanisms before this before these clinical diagnoses that are more related to the disease and can be more explanatory than or more informative than clinical criteria. And the concept of intermediate or endophenotype is very interesting because endophenotypes are basically measurable features that are present in health or disease, are measurable all the time, are inherited, and and given that they are inherited, can be directly linked to genes which are explaining the phenotype. So, and the concept of endophenotypes was basically initially developing in psychiatry. As you can see here, endophenotypes like this, for example, working memory or some glial cellular abnormalities, are features that are present in healthy individuals, but also are present in individuals with schizophrenia in this particular example. So there's definitely a genetic genetic relation, or there is a genetic relation between these endophenotypes and specific genes in or genomes. And the reasons why these endophenotypes are laterally progressing to clinical conditions is certainly unknown, but we know that the effect of the environment or potentially epigenetic factors are explaining why individuals with these endophenotypes are finally progressing to develop disease manifestations. Given these, the endophenotype concepts are very useful to characterize at least psychiatric conditions. They are being used in other diseases, including cardiovascular diseases or inflammatory diseases, and a very comprehensive transcriptomic analysis of different human diseases have identified that the interaction of these three endophenotypes can explain mostly of all human diseases. So the interaction of the inflammasome endophenotype, which has a lot of overlapping with other metabolic or other mechanistic pathways, interacting with the thrombosome endophenotype and the fibrosome endophenotype can explain most of the human diseases. In that case, I just want to give you this summary of the key aspects of the key findings from my literature review, and moving forward to tell you what the hypotheses and aims of my PhD were. So, with all this background, I think I'm hoping you share the idea that there is definitely a synergistic interaction between pathogen and host immunity, explaining the severity and duration of disease manifestations. And particularly in my PhD thesis, I explored these interactions in 4 infections: Epstein-Barr virus, Ross River virus, Coxiella burnetii causing Q fever, and dengue virus infection. So the first aim was basically using a candidate pathway gene approach to analyze the interaction of functional single nucleotide polymorphisms in genes, in pro-inflammatory genes, and the severity and duration of and the severity of manifestations in a prospective cohort in Dabo. The second aim was basically using a whole genome dengue sequencing approach to identify single nucleotide polymorphisms in dengue genomes associated with the severity of dengue infections. And finally, to perform a systematic genome-to-genome analysis, which include the host and the pathogen variations to explain the severity of disease. So chapter 1 is basically exploring the effect of specific SNPs in the NRRP3 inflammasome in the severity of acute infections. Once we are facing any acute infection or innate immunity have developed very highly conserved mechanisms to sense these infections. So one way is basically identify pathogen signatures that are obviously specific of pathogens, but are triggering pathogenic responses as fever, malaise, fatigue, or other symptoms that we are normally having after in the early stage of any acute infection. But our immunity also is sensing other kind of signatures that are released in response to these to infections. So these are not directly pathogen signatures, are basically molecules that are sensed in an abnormal environment. So we, from an immunological perspective, we have defined all these pathogen signatures as pathogen-associated molecular patterns, and all these indirect signatures of inflammation as danger-associated molecular patterns. It is interesting that these 2 signatures are triggering very similar clinical manifestations as fever or fatigue, based on the fact that common cytokines or inflammatory molecules are triggered after these DAMs or PAMs are sensed by our innate immunity. For this particular case, I focus on the inflammasome pathway, which is it's a very interesting pathway, as it is sensing PAMs and DAMs, and the final response is basically the production of interleukin-18 and interleukin-1 beta, which is translated in clinical symptoms as fever or fatigue or sleepiness, or what we are calling now as brain fog. So again, these cytokines are triggered by very diverse PAMs or DAMs, and given that both type of signatures can trigger these responses, it is reasonable to assume that any genetic variation in these pathways can be implicated in the severity of acute manifestations, irrespectively of the nature of the pathogen. So just using as examples, influenza or Epstein-Barr virus, any acute infection is characterized initially, but unspecific symptoms, which are followed after the adaptive response is developing, are followed by very specific symptoms. And these early manifestations are collectively called as the acute sickness response. And all these manifestations are actually triggered by the same cytokines, which are generally triggered by these innate mechanisms as the ones that I described for the inflammation pathway. Again, it is interesting the fact that many of these symptoms are still manifested in some individuals after infection. And we can definitely identify a link between the severity of the acute infection and these post-infectious manifestations that are harbored by many patients after infections, not necessarily only COVID, but in any acute infection can cause post-infectious manifestations, causing the same kind of protracting manifestations as fatigue or brain fog. So specifically for this part of my thesis, we focus on key genes in the inflammation pathway, which are the inflammation genes itself, CART8, which is a protein that is regulating the inflammation gene, the caspase-1, which is actually cutting the interleukin-1 beta and interleukin-18 and increase the amount and bioavailability of these cytokines in the bloodstream. So we decided to identify single nucleotide polymorphisms, SNPs, in these key genes of the inflammation pathway. But instead of selecting these SNPs in a random or unjustified way, we basically identify just those SNPs prevalent in Caucasian populations, given that the DABO cohort was an Australian cohort in individuals acutely infected by 3 pathogens. And also, in addition to this prevalence threshold, we also reduced the list of available SNPs to those with previous biological evidence of functional impact. So many SNPs are definitely present in genes, but not necessarily affecting at a transcript level or a protein level. So in addition to use this threshold of the prevalence of the SNP, we also select only SNPs that were implicated in protein in phenotypic changes as protein levels or transcript levels. So from this huge list of SNPs, almost 2,000 SNPs, we ended restricting of candidate list of SNPs to 9 SNPs in these 5 genes, as I mentioned before. The DABO infection outcome study was a prospective cohort. During 12 months, individuals were followed after they were diagnosed with acute Epstein-Barr virus, Ross River virus, or Q fever infection. And by using validated questionnaires, the severity of symptoms were measured over this 12-month period. So we basically use the clinical data captured by these questionnaires to capture severity over the time, and we genotype these SNPs in the entire DABO cohort to identify what is the role of the potential role of these SNPs in disease severity. This is just a summary of the kind of questions that were asked in the questionnaire. So if individuals had malaise or anorexia or were feeling irritable or cranky, so these questionnaires were covering different dimensions different symptoms normally presented in acute infections, including not only the typically inflammatory symptoms, but also neurocognitive or emotional or even or other symptoms as pain, for example, as you can see here. The problem was how to transform these answers or these answers into a more more useful scores or more straightforward ways to measure severity. So using principal component analysis, we basically reduce all these symptoms into 4 symptom domains. And the principal component analysis is a very common technique to reduce multiple dimensions into very few in few dimensions. So all these questions were reduced into 4 dimensions, capturing, for example, mood disturbance or fatigue or pain or acute sickness. So these 59 questions were reduced basically to 4 dimensions, measuring numbers, which gave us an understanding of disease severity. So the DIOS cohort, as I mentioned, was a prospective cohort in 484 individuals, and all the participants in this cohort were characterized in 4 symptom domains as fatigue, the domains that I mentioned before. So the problem was how to differentiate these scores, and we basically use the statistical definition to impute individuals as mild or severe cases based on their scores. So the lower quartile scores were assumed as mild manifestations, while the upper quartile as severe manifestations. And after defining these individuals in cases or controls, we match these individuals by age or gender, and perform an association analysis. So as expected, individuals with mild scores or with or controls usually resolve all symptoms, all return to the normal stage during the follow-up, while individuals with severe manifestations remain symptomatic even after many months and fulfilling criteria of chronic fatigue syndrome, which shows that the severity of the acute infection is definitely important is an important determinant of severity and protracting manifestations. And finally, for this study, when these cases and control, when the frequency of these single nucleotide polymorphisms were measured in cases and controls, we identified that 2 single nucleotide polymorphisms were associated with the risk of severe fatigue. One of these very interesting, this SNP in the inflammasome gene, is a gain of function mutation, which actually is making the inflammasome to be hyperactive and increase the production of interleukin-1 beta or interleukin-18, and interacting synergistically with this other single nucleotide polymorphism in the interleukin-1 beta. So any individual having this haplotype, has in risk to suffer severe fatigue after an acute infection, irrespectively of the pathogen. So I'm going to move now to the 3rd chapter. It's a lot of information, so I'm going to try to resume a little bit. So the 3rd chapter of my thesis was to explore the pathogen determinants of severe dengue. as other acute infections, almost 50% of dengue infections are minimally symptomatic or even asymptomatic. Almost 50% or 30% are fulfilling the typical dengue fever criteria, or even just the criteria of an undifferentiated fever. And definitely less than 10% of cases are fulfilling severe criteria as dengue, dengue shock syndrome or severe dengue. The same distribution of severity in acute infections is pretty much the same irrespectively of most acute infections. What is known in terms of the determinant of severity is that definitely a secondary dengue infection is the main risk factor to suffer a severe dengue infection. And this is basically explained by the antibody-dependent enhancement that is only reported in dengue infections, and is not and is actually protective in other infections. So suffering a secondary infection by any other infection there's less risk of suffering severe manifestations, while in dengue is the way around. Among other factors explaining the severity, in pathogen diversity is another that was already described. Unfortunately, these studies are biased by the fact that it is unknown how prevalent the secondary infections were in cohorts reporting, for example, more risk of severe dengue in Asian genotypes in dengue infections. There's also an association between age or comorbidities with the risk of dengue manifestations. So for this chapter of my thesis, I basically work in the Colombo dengue study, which is a prospective cohort in patients with acute dengue fever in Sri Lanka. And in this case, I started with 529 individuals with complete clinical data. So I use this clinical data to characterize the severity of disease manifestations. And I also I did my best to generate full-length genomes from these 500 individuals to obtain a whole genome characterization of the dengue viruses causing infection. So the most prevalent dengue serotype was dengue 2, and dengue 3 was the second one. And again, this cohort was plenty of clinical data to characterize severity. In addition to the traditional clinical criteria to characterize severity, this study had the advantage that plasma leakage was measured in individuals. So plasma leakage is was normally measured, or in low-income settings is normally measured by the concentration of hematocrit, but probably a most reliable way to measure plasma leakage is by using ultrasound to identify ascites or pleural effusion in individuals with acute dengue infection. So given that only there were no deaths in the dengue in the Colombo dengue study, and only 15 severe cases with severe dengue were identified in this study, we used plasma leakage as an outcome, as the outcome of interest. So all these laboratory and clinical data was used in addition to the definition of plasma leakage to generate, in a similar way as in the previous chapter, to generate alternative diagnosis or alternative outcomes based on the principal component analysis. So in addition to plasma leakage, we were able to generate 3 quantitative outcomes, which were lymphocytic abnormalities, symptom severity, and hepatic dysfunction. So as I was explaining at the beginning, an accurate characterization of disease severity or an accurate characterization of the host or the pathogen determinants of severity requires to include as many determinants as possible. And if we were exploring the pathogen determinants of severity, we definitely need to include common factors affecting the severity of dengue, which in this case are previous dengue infection, age, or gender. So using logistic regressions, we basically try to identify if each variation in the dengue genome was associated with the illness severity, which could be a plasma leakage or any of the endophenotypes that we generated using the principal component analysis. So after generating the full-length genomes, we basically translate these full-length genomes into amino acid sequences and remove all the amino acids that were common across all the sequences and just retain those amino acids that are variable in each sequence. And in the same way, we reduce all these amino acid variations by principal component analysis into principal components, and with the intention to include these principal components in the logistic regression. If another way to study the impact of pathogen diversity in severity is using phylogenetics, but this kind of multifactorial analysis, including more than one factor to explain severity, are not generally feasible if using phylogenetic approaches. So that's why we make these principal components, and as you can see here, these principal components were able to differentiate the three dengue serotypes, dengue 2, dengue 1, and dengue 3, in a similar way as a conventional phylogenetic approach. So we basically run a logistic regression, including age, gender, previous dengue infection, and each of these principal components in a logistic regression, and try to identify an association between these amino acid substitutions and the outcome of interest. In case of plasma leakage, no single amino acid substitution was associated with severity. And but when using the alternative outcomes as the lymphocyte abnormalities or the hepatic dysfunction, we were able to identify a mutation in the NS5 gene, which is basically in charge of the polymerase, the viral polymerase, the dengue viral polymerase. And this mutation was located in a conserved region in all this in the three dengue serotypes. As you can see in the other amino acid substitutions, these were actually located in serotype specific regions and were not capturing mutations conserved across the three dengue serotypes. So these associations are basically biased by the genetic differences between serotypes more than a real effect of a mutation in the dengue genomes. At least this mutation was located in a conserved region of the dengue genomes, of the three dengue genomes. And when running an additional analysis just restricted to the dengue 2 samples, we identify again that this mutation was implicated with severity of dengue infection. And we were also able to identify another mutation also located in NS5, which is a viral polymerase. Yeah, so I'm going to move to the last chapter, which is the host to pathogen analysis in dengue infection. Using this graphic again, the best way to characterize disease severities, including at least the host contribution and the pathogen contribution. So when running this virus pathogen to illness characterization, we were able to identify 2 amino acid substitutions in NS5. So what was pending was to explore the effect of host genome, the host genomes in illness severity, again by logistic regression, and also explore the effect of the host genomes in the viral diversity. And we plan to do this also by logistic regression. So for this last chapter of my thesis, I basically extract DNA from the same cohort and using the UK Biobank, which is a well-known and reputed array platform to characterize up to 700,000 markers, genetic markers. We were able to characterize up to 1 million genetic markers in 272 participants of the dengue cohort. Unfortunately, there was a loss of information at the beginning of the study. Blood samples were not considered, and at least 52 individuals were not didn't have DNA samples to include in the GWAS analysis. And due to the QC quality control checks, we had to exclude some other individuals and ended with this number. Again, using a logistic regression approach, we conduct a conventional GWAS study looking for associations between severe dengue and host markers. In case of plasma leakage, it wasn't possible to find any association, but when using the endophenotypes, at least 3 different SNPs were identified, but not surprisingly, these SNPs were located in non-coding regions of the human genome. I'm saying that it's not surprising because at least 90% of GWAS studies have similar findings. So are mapping single nucleotide polymorphism in a non-coding region of the genome, which is completely, it's hard to interpret if this is just a fake association, or if this single nucleotide polymorphism is in linkage disequilibrium or is regulating another gene in the human genome. So this is one of the challenges of conducting GWAS studies. So the last bit of analysis was basically to explore how the host genome is affecting the viral diversity, and the approach is basically the same as a GWAS. So we basically use, in this case, as outcomes, the amino acid substitutions in the virus proteomes, and try to associate these amino acid substitutions with single nucleotide polymorphisms detected by the UK Biobank. For this particular case, we identified that this amino acid substitution in NS5 was associated with an intronic variant in the NFAT5 gene, which is the nuclear factor activating T cells. Unfortunately, this association was below the significant threshold, but it's interesting the fact that the same amino acid previously mapped with illness severity was also mapping, was potentially mapping a gene affecting lymphocyte function and potentially explaining severe severity of disease. So in summary, I think we were able to identify amino acid substitutions potentially explaining the illness severity. We were not able to map any host SNP directly explaining illness severity by using a conventional GWAS approach, but it was suspicious that despite below a significant level, there was a correlation between an intronic variant in NFAT5 and one of these amino acid substitutions. Just to finish, NS5 is the viral polymerase, but one of their effects on immunity is also antagonize type 1 interferons by proteasome degradation of the STAT2 pathway. So we definitely need to verify these or replicate our studies, and this is in our plans. But there's some plausibility that the findings that we identify can be used can be explored in more details later. Right. So I think this is all of my presentation today. I just want to finish highlighting the fact that host and pathogen determinants are definitely influencing the severity of disease manifestations, as we try to demonstrate. Another key finding was the fact that clinical criteria could be useful for diagnostic or therapeutic approaches, but not necessarily useful to understand the pathological or pathophysiological basis of diseases. And in that case, using endophenotypes could be more informative. So we basically use pathogen determinants at a consensus sequencing level, but we are planning to conduct this type of analysis using a more in-depth viral genomic characterization to identify if intra-host variants or non-coding viral transcripts are actually explaining the severity of disease as well. There are definitely multiple factors not included in this study, as co-infections, lifestyles, epigenetics, and that we are hoping to address later. And but in general terms, I think, irrespectively of the infection, we were able to show that innate immunological responses are very important, regulating the severity of acute infections. And we are planning to replicate these methods and this approach in large cohorts, including more diverse populations. Yeah, so that's all, and thanks a lot for your time.
Thank you, Braulio, and that was a lot of work. So we may have a quick time for a couple of quick questions. Yes.
Hello. Thank you for your presentation. I have a question. So you are a clinician, or you were first as a clinician. What would be more useful for deciding the treatment if you had available the technology and everything to know the host determinant or the pathogens, to know in depth the pathogen, for example, the variant of SARS-CoV-2 that the person is infected with, or to know whether the person has SNP that predisposes it to severe disease?
Right. Well, I think this is one of the things that we are aiming to tackle in the future. If you use, for example, cancer diseases as a comparison, precision medicine is commonly used to take that kind of decisions. So we are hoping that identifying pathogen and host determinant of severity will allow us to further, as clinicians or at a hospital level, to predict disease severity or decide which treatments are more suitable for a particular case. I think at this moment, unfortunately, the data we have is very is restricted. So we have enough evidence to support clinical decisions. But we are hoping that the effort that we are conducting now will be informative for future conditions, considering that emerging diseases are now a global threat. So we are hoping to apply the same if anything similar happens again.
David.
Thank you. Great, Braulio. Thank you. Quite an involved piece of work, and you did a great job of deconvoluting a convoluted project. So thank you very much. Just before I ask my question, I just noticed that there's a Q&A there, which you may want to get to a little bit later. I was curious about the polymorphism that you saw within NS5 that is related to the virulence of dengue. Presumably, being in the polymerase, that does beg the question: where does that virulence come from? Being a polymerase, does that actually does a polymorphism induce more genomes, for example, for this pathogen to kind of replicate itself? But you were suggesting that it may be involved in a complex that may be involved in some form of degradation pathway.
Yep.
Instead, my question is, has anyone else observed anything similar in terms of ubiquitination or pull down?
Yep.
In vitro.
Thanks for the question, David. I had to remove some of the previous details of my presentation because we also check what the impact of this mutation in function of and viral fitness was when this mutation was induced. Despite this is a non-synonymous mutation, which is basically an amino acid substitution, it doesn't change any conformation in the polymerase or either affect the function of the polymerase. So that's why I directly jumped to the hypothesis that this mutation, if it's finally real, it is affecting the severity by modulating by this well-known mechanism how NS5 antagonize type 1 interferons. So there's a lot of biological evidence showing that NS5 is antagonizing type 1 interferons through the ubiquitination pathway. So we are just suggesting that this is a potential mechanism, given that the mutation wasn't affecting the stability or the function of the polymerase.
Thanks very much.
Thanks, David. So I think we are slightly over time today. There's one online question, and I'll get Braulio to answer it offline. So I think we'll wrap up for today. So thank you very much. So thank you, Braulio, and thank you everyone for coming. And we'll see you in the next seminar. Thank you.